Hi guys, today we're going to go over some of the flow cytometry. So flow cytometry itself is really just used to determine the properties of a cell sample. So cells are kind of like mixed up and suspended in a fluid, and the fluorescent markers can be added, allowing the sample to react and make sure that they're all bound properly. And then the sample itself is injected into the machine. So flow cytometric analysis uses those fluorescently marked antibodies to determine cell surface antigens, and those instruments really come in one or two flavors. So the first machine type is really analytical. It's really used to determine the cell surface protein, the intracellular proteins, kind of like their size, their granularity, and the expression. The other instrument also does this analyzing, but it does something on top of it, and it's able to really sort the cells out into particular subsets. So if we take that even further, these flow cytometers can be converted into those fluorescently activated cell sorters, or FACSs. So the instrument really just kind of sorts cells by counting their fluorescently marked antibodies, and it does so by using this beam of ultraviolet light, and then a detector which detects the fluorescent markers. So kind of as the cells pass down through this really tiny tube that really only holds one cell at a time, the cells are sorted and counted. So both the fluorescently labeled cells and the unlabeled cells are sorted and counted as they pass through that laser and detector. So this technique, of course, is really powerful, really precise. It creates really large profiles of the lymphocytes within the patient based on the amount of fluorescence within each cell. So commonly, antibodies can react with these clusters of differentiation, and these antigens are really just called CDs for short, and they're really used for specific phenotyping. CDs are really good markers for the developmental stages throughout sort of the cell's life cycle. And in immunology itself, it has a really big precedence in sort of B cells and T cells, because as they develop, they have those specific markers, and they're able to be oh, okay, well, this one's a bit younger of B cells, so it's a new one, it's a new infections that's fighting. And then also the B cells and the T cells, their functional subsets can also now be kind of phenotyped out and separated with flow cytometry. Sort of the common data that you're going to get from these results is a graph that'll have full fluorescence and then cell size, and it's going to kind of be a light scatter, and these graphs look like they have dots on them. So it's really separated again by that fluorescence marker intensity of the cells as they pass through that laser. So some of the light scatters would be sort of those distinct populations of cells. So an example that I saw was granulocytes, monocytes, and lymphocytes of a patient that was all kind of blotted out. You'd have the higher density fluorescent cells up at the top, and then the mid and the lower, and it's going kind of this really nice linear trend, and you can see that. And you can also see kind of these different wild samples. It really depends on the patient and the level of antigen binding, which then leads to if more antigen is bound to that cell, then the cell's going to be having a really more higher fluorescence. So overall, you really see that flow cytometry is really, really precise. It's really, really powerful, and it really offers that really reliable way of creating a patient's phenotype in a fairly quick method. So you're able to see what's kind of ailing them, and also you're trying to see okay, how can we properly treat this patient based on their cellular phenotype. And with this fluorescence technology, it has really increased the medical field tenfold, and it's really exciting to see where it's going to be going in the future. And overall, that's kind of your basics of the flow cytometry.